What I want to talk about in this chapter is lipids. And lipids, by definition, were originally actually hexane soluble portions of an organism. So you can imagine you take an organism, you grind it up, and you extract it with hexane, and then you look at the molecules that are in there. And what people really think of as lipids are things like the tristerenes. In biology, we talk about glycerides or glycerols and phospholipids. That's typically what people think of as lipids. But in fact, there are a lot of different kinds of compounds. Just some examples here showing you kind of the diversity of structures that you can see. Prostaglandins um, control like inflammation, bleeding. It's in response to damage to cell membranes, and then cortisone hormones, um, vitamin A, and limonene. These are all different kinds of structures that are considered to be lipids. So here's a list of the types of lipids that you'll find sort of described in Bruce. There's fatty acids, which are carboxylic acids, really, but very long chain carboxylic acids from about 12 to 18 carbons long. Um, and it goes longer. I mean, you have 22 and things like that. But typically, most of them are in the range of 12 to 18 carbons long. So there's waxes, which are actually esters of fatty acids and long chain alcohols. Fats and oils are actually the same kinds of compounds. Structurally, they're different, though. So fats are triacylglycerides or triacylglycerols that are solid at room temperature, and oils are liquids at room temperature. So that's a very vague, of course, type of definition. But the state, the solid or liquid state of the fat depends on the number of double bonds that are in the fatty acid. And then there's phospholipids, which contain a phosphate head group, and this is what we find. Triacylglycerols in cell wall membrane, but phospholipids is what we typically think about as cell wall membrane material. Sphingolipids are another type of lipid that are found in the cell wall. Prostaglandins are actually a type of hormone, and believe it was in the middle 70s when people really started studying these things intensively, and they were related. They're related to blood clotting, blood flow, those kinds of things. And it's actually when you take ibuprofen, ibuprofen is one of the kinds of compounds that sort of interferes with the production of prostaglandins. And then there's terpenes, naturally occurring alkane or hydrocarbon compounds, sort of like caraway and those kinds of things. We also have different vitamins and also some steroids. So generally, fat lipids can be classified as, and these are the same compounds we talked about before: fatty acids, glycerides, and non-glycerol. Now, remember, glycerol, if you don't remember the structure, I should say, is this, and many of the lipids contain this functional group. It's a triol. Triglyceride, so that's a glyceride, but a triglyceride has substituents at each one of these each one of these oxygens. So it has an acyl group there. So we have glycerides, and then we have fatty acids, including soap. So soap is created from long chain fatty acids. And then we have, in general, the non-glycerol or glyceride lipids, and those include the steroids. There's lipoproteins, waxes, and sphingolipids. So here we have some of the fatty acids. These are the saturated ones. I'll show you some of the unsaturated ones in a little bit. But lauric acid, myristic acid, palmitic acid, stearic acid, and arachiditic acid, uh, systematically they show the names there, but most people don't actually use those names. The only one that you see, no, actually you don't really see it very commonly. I was going to say octadecanoic acid, but stearic acid is what most people will know it as. And if you, sorry, little side thing. If you look at the melting points increase steadily with fatty acid chain length, and that's because of van der Waals interactions, sort of the induced dipoles that occur between nonpolar substituents. The carboxylic acid has some role in that, but really it's that length of the fatty acid chain that's important. Now let's take a look really quick at some of the unsaturated ones. So we'll look at palmitoleic acid. That's the first one. That's C16, and we'll look at palmitic acid. And notice the big difference in the melting point, and that has to do with the double bond that you find in this position here. Now, if you look at all the acids in terms of degrees of unsaturation, so for C18, that's stearic acid, that's oleic acid, linoleic acid, and linolenic acid. That's increasing number of double bonds. And if you'll notice, the double bonds all on the 9th carbon. So we'll go like this, right? 1, then 2, 3, 4, 5, 6, 7, 8, and 9. And it does that for every one. And also, you should notice that if you look between the double bonds, so for linoleic acid, for example, or linolenic acid, this carbon is sp3. So at first glance, these things look conjugated, but in fact, they're not conjugated molecules. But that sp3 carbon turns out to be very susceptible to radical reaction attacks, and so we'll talk about that as well because of the resonance that it has with the double bonds on both sides. So arachidonic acid is the precursor to the prostaglandins, and it's closely related to arachiditic acid. But notice the melting points again with increasing number of double bonds. So for example, if we look at oleic, linoleic, and then linolenic acids, even though they're all C18 and have very similar structures, right, the increased number of double bonds decreases the melting point for these compounds. Oh, one other thing that I wanted to mention that I forgot to, so I sort of restarted my recording here, is that the double bonds that you find in naturally occurring fatty acid chains are all in the cis configuration. So you'll notice in, for example, palmitoleic acid, this bond is in the cis, sorry, I'll use this as a pointer. This is in the cis configuration. And then in linoleic acid, it's cis and then sp3 and then cis, and then linolenic acid, it's cis sp3 cis sp3 cis, like that. And then arachidonic and EPA, that's eicosapentaenoic acid, pentenoic acid, with the 5 double bonds, right? Those are all in the cis configuration, and every double bond is separated by an sp3 carbon. So I don't know what else to call this, so I just called it fatty acid factoids. Naturally occurring fatty acids have an even number of carbons, kind of summary of what we've talked about. We have polyunsaturated fatty acids that have more than one double bond. Now we have monounsaturated and we have saturated. So when we're talking about saturated and unsaturated fats, that's actually where those terms come from. The double bonds all have a cis configuration, and they're all separated by one methylene group. That's an sp3 carbon, and as you increase the number of double bonds, generally speaking, you decrease the melting point. So when we talk about triglycerides or triacylglycerides, what we're talking about is glycerol, which I showed you earlier, and it being bonded through an ester linkage to an acyl group, or I should say bonded to an acyl group. So remember, an acyl group is just a C double bond O with an R group next to it. So when you have a fatty acid and you create a triacylglycerol out of it, or a triglyceride out of it, the R groups don't all have to be the same. Now, depending on the composition of the R groups, if it's all saturated, then most certainly it will be solid at room temperature. But then depending on the number of double bonds in the R groups, right? Again, the R groups don't all have to be the same. You get varying degrees of melting points. And so the more double bonds you have, polyunsaturated, for example, fatty acids, in a triglyceride will make it liquid at room temperature. And again, that's the only differentiation between fats and oils. So if you look at a fat, and I'll be honest with you, this is a picture out of Bruce. They're not this perfectly aligned. If you try to simulate this, it's really hard to get it to simulate like that. But that is the idea, that when you have fatty acids and you make a triglyceride, if they're all saturated, they can become these cylinder-like objects. So imagine that you're trying to pack these things in, right? They have the ability, so I'm going to do it like this, as cylinders like this, all right? They have the ability to very efficiently pack cylinders. And interestingly enough, if you take stearic acid and you put it on water, it will actually stack on the surface of the water with all the carboxylic acid groups in touch with the water, and it makes actually this like a hexagonal array of fatty acid chains, all linear, sticking up from the surface. It's one of the fun labs that occasionally we do where we can calculate Avogadro's number from the size of an oil drop on water. It's actually stearic acid drop on water. All right, so anyways, yeah, so they line up like that. And the reason that's efficient is because, you remember, with induced dipoles, the surface area contact is what's important. So these molecules can come into perfect contact with each other if they're cylindrical like this. So this would be a representation of something that would be a fat. Now, if you put one or more, okay, and these are probably oleic acids, I think. I didn't count them, and I didn't actually look at them that close. But because of the cis double bond in it, what happens is there's a kink in the molecule, and if they're all the same, the kink occurs in the same place. So what that does to the molecules, remember we talked about this cylindrical packing, right? Like this. Fatty acids that contain unsaturated, fatty acids that contain unsaturated, that are unsaturated, can't pack like that when they're in a triglyceride. So that would be trying to pack something that looks like, instead of straight down like this, it looks like this, and then like this, and then, it just doesn't pack together well. And again, the importance of the packing together is that the surfaces can come into contact with each other, and then those induced dipole forces can be relatively strong. So when we're talking again about fats, right? You can actually hydrogenate the double bonds and make them either monounsaturated or saturated. So you have a polyunsaturated fat, right, and you subject it to hydrogenation, then you can make it into a saturated fat. And that's how, in fact, we get things like Crisco. Crisco is actually from cottonseed, and it's actually an oil before it's treated. And then they hydrogenate it, and by removing the double bonds, what happens is its melting point goes up, and it becomes solid at room temperature. You might be asking, like, why in the heck would you do that? Because isn't it bad for you, right? This is actually goes back to the days before refrigeration. So imagine that you want to store something, right? Well, a couple of things that you have to consider. I mean, if it's a liquid, it's not as easy as to store as a solid, right? So you can store solids in a different place. So yeah, so before refrigeration, this is one of those things that people thought about. The other thing, though, is by removing the double bonds, it actually becomes more shelf stable. And so people tended to store things at room temperature, or they had ice boxes and they'd have to buy ice, but they didn't usually like keep their lard or their fat in there. And so the idea is if you can make it more shelf stable and keep it as a solid, those are both positive things. And so that's what they've done. They did originally, and now actually I think Crisco is not actually made from cottonseed oil anymore. I think it's actually made like out of soybean oil or something like that, something that's common and easy to get. So this slide deals with a little bit of the reason like why we do hydrogenation of oils. So think about this is a segment. Oh, sorry, let me use this pointer. This is a segment of an unsaturated fatty acid. Remember, this carbon in the middle of the two. This is a methylene group or an sp3 carbon. If you generate a radical, let's say with air or through some other method, it actually starts a chain reaction. This hydrogen is actually reasonably easy to pull out because um, this carbon's radical is stabilized by resonance in both directions. Now, the resonance contributor with the conjugated double bonds, this one's the one that's more stable, and it'll actually radicalize, I guess, if you want to put it that way, oxygen, right? So to get the oxygen, basically, the oxygen uses one of its radical oxygens to combine, and then you form this peroxy radical. Now, over a period of time, this breaks the molecule in half, and you end up with these shorter chain carboxylic acids. And when a fat goes rancid, that sort of sour smell that you smell is actually the carboxylic acid that's created after this initial oxidation of the fatty acid chain. So you can imagine if you made something like Crisco, it wouldn't be nearly as susceptible to this because you don't have nearly the degree of unsaturation or double bonds in the hydrocarbon chains. So now we come to actually stuff that's important for cell membranes in biology. And these are known as diglycerides. They're diglycerides because you have two glycerol groups here that are acylated. So you have your two fatty acid chains. And then what you do, so that's the diglyceride part. And then what you do is you modify the group here with a phosphate head. And this is actually the major component of cell membranes. Okay. There's a couple of different kinds of phospholipids or phosphoacylglycerols. I find it's just easier to say phospholipids. There's a couple of different kinds of phospholipids. There's cephalins, which we find a lot in nerve tissue. Lecithins, again, a common cell wall. And then phosphatidylserine is another common structure that you'll see. These are all considered to be phospholipids. So we're going to talk a little bit, and this was actually one of the labs we typically do. We actually make soap, and soaps are actually fatty acid salts created from fat. So you take, for example, lard, which contains a high amount of triglyceride as well as other things, and then you subject it to hydrolysis by sodium hydroxide. So if you remember, sodium hydroxide is very good at breaking ester bonds, in part because it forms this carboxylate ion, from the acid-base reaction of the sodium hydroxide with the carboxylic acid that's released as a fatty acid chain in this case. So this process, when we use sodium hydroxide, is called saponification. And you can actually do, this is kind of a home thing you can do almost. You could take lye, which is usually sodium hydroxide, but occasionally you'll find things with potassium hydroxide in it. But sodium hydroxide, and then you can take a fat like bacon grease, and then you can mix the sodium hydroxide into it, and then what you end up with is these fatty acid salts. Now you have to be a little bit careful when you do this at home, because there is that possibility that you add too much sodium hydroxide and your soap ends up being really, really harsh and basic. But this is actually, again, like one of those things, if you can look up a recipe online, it'll tell you exactly how to make soaps at home. And people say the soap that you make out of bacon is actually really good. I used to have a lady back, I don't know, 10 years ago, used to come by and collect bacon grease from me, and she wanted us to save it, and I kind of did it a couple of times, and I got tired of saving bacon grease. I don't eat that much bacon to start with, that I make that much grease. But yeah, this lady was going house to house collecting bacon grease and making soap and selling it. So anyways, soaps and detergents, right? Hydrolysis reactions of esters is, in essence, a triacylglyceride is just a triester, right, to form glycerol and fatty acid salts, which are soaps. So phosphoacylglycerols or phospholipids is what we usually learn them as. Are the major constituents of cell walls, and they form what's called a lipid bilayer. And the idea of a lipid bilayer is that it allows for the most stability of the lipid component, the hydrophobic components, as well as the hydrophilic components. So in a cell wall, we have a phospholipid like this phosphoglyceride that we're showing here, and there's a polar head group on it, and this is the part that likes to orient towards the aqueous phase. The nonpolar tails tend to orient towards the nonpolar portions, and so what happens is, in a cell wall and a lipid bilayer, what we get is two layers. The tails are all lined together, and the heads are all lined together, because the water, like H2O, would be out here like this, or inside the cell there's also H2O. And so that minimizes the energy of the lipids in the cell wall. There are a lot of other constituents of cell walls, including things like arachidonic acid, cholesterol, and there are both saturated and unsaturated chains in the alkyl portions of the phospholipid. And this sort of modifies the fluidity of the cell wall. So cholesterol in general is something that helps to make makes the cell walls more rigid, right? And then again, unsaturated fatty acids in the cell wall have the opposite effect. Another interesting structure, and this is actually not covered in your book, it's called a micelle. I don't know if that link is good anymore. I didn't check it, but I downloaded the PDB file, is what it's called, so that you could take a look and see what a micelle looks like. So let me find it here real quick. So here's a micelle, and this is, I think, a choline at the end of the phospholipid. So these are, let's see if I can zoom in here. I try to see just one in isolation. But what you see is, in this structure, the polar portions are all on the outside, and then the inner portion of this structure contains all the nonpolar parts. You can actually, in this type of file, include water molecules and either show them or not show them. So if you show the water molecules here, let's do this again real quick. Pause this. This is actually what it looks like with the water molecules present in the structure. And you can see how they completely surround this micelle. Okay, so micelles are interesting because they're actually looking at them for things like drug delivery. You could take a nonpolar drug and then incorporate it into a micelle formation, and then it can float freely in the bloodstream. And then when it finds a cell wall, it can incorporate the contents of the micelle into the cell wall. And then they're also looking at modifying the outsides of these, so they have specific molecules on the outside so that they're attracted to or attached to specific target molecules. This is actually a bilayer reconstruction using PDB files. And so I always thought it was interesting. One of the things that I always thought when you're looking at lipid bilayers, so you can see the polar portion at the top and you see non-polar portion at the bottom. In this bilayer portion, non-polar part in the middle and a polar part at the bottom, this pretty clearly shows, well, it's not exactly, that you have two sets of molecules, top and bottom, and then there's this sort of layer where the tails interconnect. Now, I don't know if you can see this or not. There's actually another structure on the inside of this, so what I'll do is I'll change a few settings to highlight that. So what I had to do is I ended up changing everything to wireframe and then changing all the lipids to one color. And now what you can see is in here, this is actually a protein structure. And you can see alpha helices and things like that in the structure. And this is actually a protein channel that they imaged. And so when you're looking at this, you have to think about, that's the environment. I didn't talk about this when I did the amino acids chapter because it didn't really have that much meaning. But now we can talk about it a little bit more. When you have channels that run through lipid membranes are predominantly hydrophobic, and when these proteins get inserted, the hydrophobic portions of the protein are all on the outside, or predominantly on the outside of the structure. I believe this is an ion channel, so the hydrophilic portions are in the middle. Now, I don't have any, there's no way to show whether or not there's water molecules in here, but this is actually a protein structure, and this is what proteins look like when they incorporate into a cell wall. They sort of transverse the lipid bilayer. The other thing I was going to point out, because it was a misconception that I had for many years when I was studying biology, is that I always thought that the lipid bilayer tails crossed more, but in fact they only butt up against each other, and there's a very small region of overlap there. Former students of mine have done research in this area, and what they've in their part of their research is they've used very cold conditions to rapidly freeze the cell membranes, and then they actually crack them open, and you can find the proteins sticking in and out of the two layers of the cell membrane. So these are separate enough entities that when you freeze them, you can fracture it and separate the layers of the lipid bilayer. Another interesting structure, a structure that I like, is if you take a bilayer and you sonicate it, so you use one of those cleaning things that makes the high frequency sound, you can sonicate it. You can actually take the lipid bilayer and break it up, but as an entity, the bilayer doesn't like its ends exposed, so it forms these circular things that are called structures that are called liposomes. And the liposomes is like a mini cell, and you have interior portions that contain an aqueous portion, and the exterior part is also exposed to the water, and then you have the nonpolar lipid bilayer in between. So let me show you just a little bit of this video real quick. It's kind of corny, but I think it's really neat. This is I'll show you the link here in just a second. Or you can look up, it's actually called The Good, the Bad, and the Ugly Optical Tweezers. And what they do is they take lasers, and you can move objects as small as liposomes. So this is actually a yeast cell. Right? I'm going to just skip a couple of sections here. They're moving a bacterial cell. And this is actually a liposome. So that structure that we were talking about, you're able to move liposomes. And again, transport. Liposomes are transporters, so you can put things inside of them and move them around. And they're sort of setting up this sort of drama with the liposomes. And just for a second, let me explain to you how this thing actually works. So this cone actually represents laser light. And the sphere, which is the liposome, allows the laser light to be focused through it, and then as it comes through, you have to remember laser light is actually pretty much vertical. When a laser light comes through the liposome, what happens is there's a lensing effect. And so light comes up through the side here and then exits over here, and light comes through here and exits over here. So this cone is showing the sort of trajectory of the laser as it leaves. Well, one of the things we don't talk about very much in chemistry is that light has momentum. We mention it in the quantum mechanics section, but that means it can actually move objects. And so these liposomes are so small that the laser light's momentum can actually move them through solution. So I've updated the slides. This link right here, this is the author of the video, and this is the link to the video. So if you want to watch it, that's where you would go. Some of these images actually came from this link, but apparently that link is no longer good, so I went and found this other one. Okay, so the next thing to talk about is sphingolipids. So sphingolipids are found on in the cell wall. So they're membrane components. They only have a single tail. You'll notice it's not completely. It looks like it could be derived from glycerol, but it's not derived from glycerol. It has. Oops, sorry. It has a hydrophobic chain, again, just like all the other lipids do. And then it has this very polar head group. So that's the section that sticks outside of the cell membrane. So if you think of a cell membrane as being a bilayer like this, the sphingosine tail would go in, and then they would have this double bonded component, the double bonded components inside, has the CHOH and the NH2 and the OH on the outside. And the function of sphingolipids is apparently to provide some structural stability to the cell membrane as a sort of protective function. So there's several different kinds of sphingolipids. There's sphingomyelin, and there's sphingosine, right, and then sphingomyelin, which is a modification of sphingosine, and there's this glucocerebroside. So sphingomyelins are a common component of the myelin sheath of a neuron. So in your body there are neurons. I don't know how much of this you guys know, but you have neurons which conduct electricity, and the myelin sheath is used to help really facilitate electrical conduction over long distances rapidly. And so motor neurons in our body are all myelinated. And so when we have problems with our myelination, then we have problems moving, basically breathing, all kinds of things. So it's associated with a number of different kinds of diseases. But yeah, sphingomyelin, and the cerebrosides are types of lipids that are found in cell walls and have different kinds of functions. Now, prostaglandins. Sorry, my mic was off. Prostaglandins are synthesized from arachidonic acid, and so there's a bunch of different types. The name format, there's a PG, and then there's a letter with the letter indicating like the type of ring that's created in the prostaglandin. You don't need to really know this, but again, the formation of prostaglandins is done through a cyclooxygenase, and you'll notice that it does the same thing that causes fats to go rancid. It incorporates an oxygen, creates this peroxy radical, and then forms this ring. So we have this peroxidase, which is an enzyme, which then produces a number of different intermediate prostaglandins, and also all of these thromboxanes, prostacyclins, and prostaglandin. So I made this slide a few years back. I just kind of wanted to point a couple of things out. So prostaglandins get made in your cell wall, or made from your material in your cell wall when a cell wall gets broken. and that releases arachidonic acid. And then there's a lipoxygenase, which then leads to leukotrienes, which are implicated with asthma, cystic fibrosis, irritable bowel syndrome, and psoriasis. And then there's an enzyme called the cyclooxygenase. And when I made this slide, I think there were, like I said, there were three. Those are the three I listed. But there's actually a number of others that exist, and they all have sort of different kinds of functions. Prostaglandins are part of the inflammation and pain cycle, but thromboxanes are part of the blood clotting cycle. So you get these different enzymes that produce these different kinds of responses. Well, as it turns out, the enzyme cyclooxygenase has a receptor for arachidonic acid, or not a receptor, it has a pocket for arachidonic acid, and it then produces the radical that causes the formation of the formation of this intermediate. Sorry, I keep pointing with the wrong thing. This intermediate. So the cyclooxygenase is a very important enzyme in that process, and you can actually fit an ibuprofen molecule in there. And so that's kind of where ibuprofen and aspirin and a number of other analgesics, painkillers, and anti-inflammatory drugs have their site of action. But it also, it turns out, has the same effect. There's a cyclooxygenase in your stomach lining that helps to produce the mucus in your stomach lining. And so when you take these non-steroidal anti-inflammatory drugs, things like aspirin and ibuprofen, they will block that, and that's why you can get ulcers if you take too much. So I sort of listed a bunch of different drugs, but the two that to pay attention to are like this. This is the closest one to look at. And I don't know that I've ever seen this marketed, but it's a type of cyclooxygenase inhibitor. But you look at the structure of naproxen, right? Naproxen is a very common one that people use. Indomethacin, again, naproxen's here, indomethacin here. They all have this sort of structure that sort of blocks the arachidonic acid from getting in there. And then ibuprofen does the same kind of thing. That's the shape of arachidonic acid. You can see all the double bonds, the red regions that are in the arachidonic acid, and this is the shape of the pocket that the arachidonic fits into inside the enzyme. If you look at naproxen, you can see how naproxen might work, because this part of the molecule, right, this is arachidonic acid, this part kind of looks like this. It's actually twisted a little bit. I should have probably turned it a little bit more, but it has this very polar part for the oxygens, right, and then it has this sort of hydrophobic nonpolar part that fits into that pocket. I don't expect you guys to remember this part, but your book has the 28B book has quite a bit of detail in it. And remember, I told you the letter PG as prostaglandin. There's A, B, C, and D, right? The letters indicate the type of ring that's formed in the structure. So there's some examples of PGE1, PGE2, and PGF. Terpenes are interesting because they're made up of 5 carbon isoprene units. Now, an isoprene unit looks like this. It's a 5 carbon iso, right? This is the iso. 5 carbon iso structure. And it turns out in the biosynthesis of terpenes, a number of molecules of this shape are joined to form different kinds of products, natural products. So terpenes are a group of compounds that if you can divide it, if you look at a molecule and you can divide it into subunits of these 5 carbon shapes, then we can classify it as a terpene. So it's called the isoprene rule. So if you look at, for example, this molecule on the right, It's a tail to head connection. This is a 5-unit isoprene unit, and this is a 5-carbon isoprene unit over here as well. Here's an example of a molecule. This is actually menthol, okay? And if you look at this, you can actually divide it up neatly into isoprene units. So let me grab a red pen, and you can draw a line across here, and you'll see this is 1, 2, 3, 4, 5 in that iso shape. And this is the other one, 1, 2, 3, 4, and 5. So that line splits the two isoprene units, and so we know we have what we call monoterpenes, sesquiterpenes, diterpenes. Sorry, I can't even say the rest of them. But anyways, there's these different classifications of terpenes. You don't need to know like what the different classifications mean, but in these terpenes there are 2, 3, 4, 5, 6, and 8 isoprene units. So here's another example. This is celery oil. And let's see how you might break it up. So what I want you to do when you try to do this, focus on where you see these potential iso groups and try to identify where those are, because that'll give you ideas about how to split up the rest of the molecule. So I'm going to pause this and I'll split it up and then we'll count it up, okay? So there's that one. That's how that splits up. That was more difficult actually than I thought it was going to be. And the reason is you have to cut where I put the red line. You have to cut it that way. If you cut it the other way, I don't think I was able to split it up into units of 5. But you can see the different colors and how I highlighted them. So again, this would be 1, 2, 3, 4, 5. 1, 2, 3, 4, 5, and then 1, 2, 3, 4, 5. So it's not just sufficient that you count the carbons, but you also have to identify the isoprene group that's inside the terpene, a potential terpene structure. So another important terpene is squalene. It has 30 carbons in it. It's actually produced from two 15-carbon precursors, and then it produces this 30 carbon or 6 isoprene unit terpene. It's important, and we'll talk about this a little bit later, in the formation of cholesterol. It's the one of the precursors to it is the precursor to cholesterol. Now, lycopene and beta carotene are important because they're found in things like tomatoes, carrots, this bright fruit, and that part of the coloring of the fruit comes with a high degree of conjugation throughout the molecule. But if you take beta carotene and you split it in half, you get 2 molecules of vitamin A, which is what we associate with good vision health and things like that. So just so you see this, this is actually vitamin A retinol, but I mean to zoom like that. You can see that this structure, right, if you split it in half, so we're talking about 15 carbons, I believe. Let me double check. Yeah, so I counted, it's actually 20 carbons. I don't know where I got, or 40 carbons. So half of it's 20 carbons. That's what gives us retinol, and that's vitamin A. And then this is the molecule in the eye whose shape changes in response to light, and we're able to detect the change with receptors in our eye that allow us to see light and color. So the vitamin A gets bound to this opsin protein. Sorry, I hit that maybe a little bit too soon. It has an amine group on it, and then it forms an imine type bond to the opsin protein, right? And then visible light takes rhodopsin from the cis form to the pointing with my finger, cis form to the trans form, and that's how we detect the light. And then it gets recycled, the bond gets broken, and it gets recycled and then reused again. So the last thing to talk about is steroids. There's a 4-ring system, right, that makes a steroid ring. And like it says here, many hormones are steroids. And hormones, really all they are is they're chemical messengers that act at a distance, right? So neurotransmitters, we talk about neurotransmitters sometimes. Neurotransmitters are short-range communication. Hormones are long-range, generally longer-range communications. The rings are A, B, C, and D, and they're transfused, is what we say. It doesn't mean not like transfusion, like you're thinking, but the where the rings meet, the CH3 and the H, for example, these are trans to each other, and that actually has a important implication, I guess, in that it makes the rings flat. So if you were to cis fuse, you see here on the right, if you were to fuse it into cis configuration with the CH3 and the H, that would require that the ring be pointed down. So all the naturally occurring steroids have this transfusion, I almost hate to say the word, in the rings so that they lie flat like that. Just kind of a side note, I wanted to show you, I was talking about squalene being the precursor. If you count the number of carbons in squalene, right, it's I think a sesquiterpene, 1, 2, 3, 4, 5, 6. So it's six units of 6 isoprene units. And there's an epoxidase reaction that causes an epoxide. Remember when you have an acidic situation, the epoxide will be attacked at the more substituted carbon. And then it's a series of electrophilic addition reactions that zips that whole thing up at one time. So, it's pretty fascinating that it can do that all by itself. But yeah, so anyways, that's what happens. And then you have this protosterol, and then lanosterol, and then you have cholesterol. Like it says, 19 steps. These steps are enough to look at, right? But I just wanted to get you the idea that squalene, right, is what is the precursor of cholesterol. Now it's a steroid, right? Cholesterol is a steroid. It has this shape. And then there's a lot of naturally occurring steroids, cortisone and aldosterone. A lot of these have very different kinds of behaviors. They control minerals like potassium, but they also help with healing and cell growth and things like that. And then there's also synthetic steroids, and really the thing about synthetic steroids is they're designed to mimic the shape of steroids like cortisone or aldosterone. You don't need to know any of these, and here's a couple other ones just to look at them. You don't really need to memorize any of these, except you should know the structure of cholesterol. You don't need to know the stereochemistry of the CH3s and the OH groups. That maybe over time might be something that's important for you, but really just to know this cholesterol ring shape, this one here. The numbering's really not that important, but the cholesterol ring shape is one that you should remember because you'll see it a lot in the future. And so that sort of brings us to the end. Again, if you have any questions, feel free to send me an email or send me a text and let me know what your questions are.